What are the things that you need to be thinking about in terms of what a plant needs to do photosynthesis? You think of it in terms of the reactants, the molecules that it needs to function in terms of doing photosynthesis, but you can also think of it in terms of the cellular machinery that it needs to do photosynthesis. So what are these things that cells need? Both in terms of reactants and then things inside of themselves.
Needs CO2.
Okay, so you need carbon dioxide. Where's carbon dioxide going to be found?
From the air outside.
Okay, so it's going to be found from the air outside. So let's add some epidermal cells here. And what guards the opening to the inside of the leaf?
Guard cells.
Guard cells. So we have these enlarged cells that basically form the opening to the stoma. So we have CO2 outside the leaf. Where else do we have carbon dioxide? Yeah.
Inside the stomata.
Okay, not inside the stomata. I mean, that moves through the stomata, but it's going to be where? In these air spaces within the leaf. So we also have carbon dioxide in here. What else do we have in there?
Water.
We have water vapor. That's both inside and outside the cell. What else do we have?
Oxygen.
We have oxygen. Okay. We have nitrogen and other stuff, but these are the things that actually matter for photosynthesis. So now what does the cell have in terms of its machinery for capturing carbon dioxide?
Is it Rubisco?
Yeah, so these cells have Rubisco in them. And what does Rubisco do again? Yeah.
Requires less amount of CO2 to get a reaction.
It requires less amounts of energy in order to get a reaction. So it's an enzyme. So that's the kind of the definition of an enzyme. Good. What else do you know about Rubisco? What reaction does Rubisco catalyze? What reaction does Rubisco force to happen? Yeah.
Like carbon. It like is a catalyst to carbon reaction, right?
Yeah. So it adds carbon dioxide to a 5-carbon compound that is here inside the cell. So inside the cell, there's this 5-carbon compound called RUBP. And what Rubisco does is it takes carbon dioxide and adds it to that 5-carbon compound. That gives you a 6-carbon compound that then breaks down into 2 3-carbon compounds. Okay? So that's just the details of kind of carbon capture and the beginning of this process by which we take those carbon dioxide molecules and convert them into a glucose molecule. So as the cell is doing photosynthesis, what happens to the concentration of carbon dioxide in these air spaces? It goes down. It goes down to the point that it becomes low enough that what starts to happen? Because Rubisco not only adds carbon dioxide to RUBP, but it also adds what?
Oxygen.
It also adds oxygen. That's what the O is. It's a carboxylase and an oxygenase. So remember those. As carbon dioxide concentrations go down, what does Rubisco start to do?
Starts to use the oxygen.
It starts to interact with oxygen, so it adds oxygen to RUBP, and that slows down the rate of photosynthesis. So what does the plant leaf do to compensate for this?
Has to open up and get more CO2.
So it has to open up its stomata, so the guard cells are going to change their shape. Open up the stomata, CO2 will flow in and restore the concentration of carbon dioxide that was there. And then what else is happening while those stomata are open? Yeah,
Losing water?
you lose water. And so water flows from high concentration to a low concentration, flows out of the leaf. Once CO2 has been restored inside the leaf, these guard cells swell back up and close off that stoma, and then the plant can go back to doing photosynthesis with a nice, good supply of carbon dioxide. This is not a problem in an environment where plants can just open their stomata up anytime they want and get more CO2 in. So this works good in cool, moist regions. And so in cool, moist regions, you tend to find a lot of C3 plants hanging around and growing and doing fine. In hot, dry regions, though, the more time you spend with your stomata open, the more CO2 you're getting, but also the more water you're losing. And so if you're a C3 plant in a hot, dry environment, you tend to lose a lot of water in your goal to get enough carbon dioxide to do photosynthesis. So C4 plants have come up with a different way of doing photosynthesis. And instead of photosynthesis all occurring in the mesophyll cells, where does photosynthesis in C4 plants occur? Yeah.
In bundle sheath cell, right?
It occurs in the bundle sheath cells. I'll draw them in a different color. But initial carbon capture happens where in C4 plants?
The carboxylase?
Okay, so that's the enzyme that is used. Where's that enzyme found? Is it found in the bundle sheath cells?
Mesophyll?
It's found in the mesophyll cells. So here we have both the mesophyll cells and we have the bundle sheath cells. We still have the epidermis down here. We still have big guard cells guarding the stoma. We have carbon dioxide, oxygen, and water. We have carbon dioxide, oxygen, and water out here. What's the enzyme that is in the mesophyll cells? Remind me again.
Pep carbon.
Carboxylase. So carboxylase in here is another enzyme. It's an enzyme because it has this ASE at the end of the word. That's how you know it's an enzyme. But this is a carboxylase. It doesn't have an oxygenase function, so it only interacts with what molecule? Carbon dioxide. It only interacts with carbon dioxide. It's not going to touch this oxygen that is out here. So when this mesophyll cell goes to capture carbon, it's going to capture carbon. It's going to add the carbon dioxide to this molecule called PEP. It's phosphoenol pyruvate, but you don't need to know that. You can just learn it as PEP. It's a 3-carbon compound. Plus this 1-carbon compound leads to a 4-carbon intermediate, which is why this is C4 photosynthesis. That C4 intermediate, you may or may not remember, is malic acid. But you should remember that. What happens to this malic acid once it's formed? This is the 4-carbon compound. Where does it go in C4 photosynthesis? And this is the brilliance of C4 photosynthesis.
Isn't it used as energy? Reused as energy?
It's not used as energy. It's basically a way of getting the carbon dioxide that you fixed here to someplace else, where do you want that carbon dioxide to ultimately be?
In your bundle sheet cell.
You want it to be in the bundle sheet cells because what enzyme is in the bundle sheet cells? Not carboxylase, but Rubisco. Rubisco is in here. So malic acid gets transported into the bundle sheet cells. In the bundle sheet cells, malic acid loses its carbon dioxide molecule that is then picked up by Rubisco, which then adds CO2 to RUBP to form this 6-carbon compound that breaks down into 2 3-carbon compounds and goes through the same process of making glucose that the mesophyll cell normally does. But the mesophyll cells don't go through that process in C4 plants because they don't have Rubisco in the mesophyll cells. That Rubisco only occurs in the bundle sheet cells. And I've drawn the bundle sheet cells in a different color because they are different in that they have a really strong barrier that goes around them, a cell wall that is covered in lignin, and this prevents any gases from diffusing into it. So CO2 can't diffuse directly into it, water can't diffuse into it, oxygen can't diffuse into it. So it's this protected space for Rubisco to do its thing without the complication of ever interacting with oxygen. So as a C4 plant is doing photosynthesis, what is happening to the carbon dioxide levels in the leaf spaces? Dropping, 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 dropping. How far do they drop? Before I just wrote CO2 in little small letters, indicating that there's less of it. How far does it drop in a C4 plant? How far can it drop in a C4 plant?
Does it drop all the way?
It can drop all the way to nothing. It can use up all of the CO2. Why does it not have to worry about the fact that now there is no CO2 and a lot of oxygen in the leaf spaces?
It's able to capture it from the air quicker. Right?
Well, that's the next step. But why here the plant, if a plant could worry, which plants can't worry as far as we know, but if a plant could worry, as the carbon dioxide levels drop in a C3 plant, the plant has to get worried about CO2 levels dropping because what starts to happen? Rubisco starts to interact with what? Oxygen. So why does a C4 plant not have to worry about the fact that there's no carbon dioxide in its leaves?
Because it doesn't interact with oxygen.
It doesn't interact with oxygen. All that oxygen just hangs out there because PET carboxylase doesn't give a rat's ass about oxygen. Never going to interact with it. So the C4 plant can totally deplete all of the carbon dioxide in its leaf spaces. Basically draw it down to nothing. Use all of the carbon that is in there. Then at that point, there's a really steep concentration gradient. There's no carbon dioxide in here. There's some carbon dioxide out here. The guard cells change their shape. Opens up the stomata. What happens? CO2 rushes in. Water flows out. But because of that really big difference in concentration, the CO2 diffuses very quickly. The steeper that concentration gradient, the faster diffusion occurs. So if you're going from a certain amount of carbon dioxide to a little bit more carbon dioxide, diffusion is very slow. When you go to from no carbon dioxide to a lot of carbon dioxide, that carbon dioxide flows very quickly because of the differential in concentration. As a result, carbon dioxide is restored very quickly so that this plant can leave its stomata open for a shorter period of time before it closes them down, causing them to lose less water. As a result of this, these types of plants do better in hot, dry environments because they open their stomata less often, and when they do open their stomata for shorter periods of time. And it's all because these plants have evolved to protect Rubisco in this oxygen-free environment that is the bundle sheath cells. So it doesn't matter how high the concentrations of oxygen are relative to carbon dioxide in the leaf, they're protected from oxygen. They're never going to interact with oxygen. And the enzyme that is responsible for initial carbon capture are out here in this oxygen-filled environment, but it's a type of enzyme that doesn't interact with oxygen. That's the fundamental difference between C3 and C4. The site of carbon capture is in the mesophyll cells, and the site of making glucose is in the bundle sheath cells. In C3 photosynthesis, carbon capture is in the mesophyll cells, and at the same place, the plant makes the glucose. All that occurs in the same spot. Now, the result of this is that it takes a little more energy to do this, because you have more steps, and each chemical reaction requires a certain amount of energy. So C4 plants use a little bit more energy, but the advantage that they get from that is that they don't ever have to interact with oxygen. And so their photosynthetic rate never slows down. The only thing that slows down the photosynthetic rate of a C4 plant is the supply of carbon dioxide. Is it getting enough carbon dioxide? You don't ever have oxygen competing for access to your cellular machinery for making glucose. It just doesn't happen in C4 plants. Did any of you guys think of an analogy that kind of gets at kind of how C4 plants do photosynthesis? No? A couple of years ago, a student asked me a way of kind of trying to remember this. And I came up with an analogy. You can adopt it if it works for you. You can get rid of it if it doesn't work for you. You guys know what an analogy is, right? Something is like something else. I'm going to propose to you that C4 photosynthesis is like Rapunzel. Who can tell me the story of Rapunzel? You guys read fairy tales when you were a kid? Yeah.
Yeah, she's trapped in a tower and she lets her hair down, the prince can climb in and can save her, something like that.
Yes, that is exactly it. Rapunzel, a beautiful princess maiden. We'll draw her with a skirt because we are gender rigid people. Rapunzel, she is in a tower. What is surrounding the tower? This is a knight. I can't draw a knight's helmet. Anyway, this is a knight. What's around the base of the tower? You guys remember from the story?
Isn't it brambles or something like
Yeah, these deadly brambles. So, weeds with spikes coming off of them. Spiky protection. Such that the knight, of course, what does the knight want to do?
Get to the princess.
Well, of course the knight wants to get to the princess. We're not going to talk about why the knight wants to get to the princess, but I think we all know. So the knight wants to get to the princess, but the princess is sequestered up in this tower, protected from any knight that might be interested in the princess. The knight comes, tries to get to the princess. Rapunzel grows her hair long because, of course, Rapunzel also wants the knight to make contact with the princess. It's amazing how fairy tales are not very wholesome when you think about it a little deeper. But we're not talking about that. She grows her hair long. What does the knight do? Latches onto the hair, climbs up, but gets partway up, and what happens to him? He falls to the earth, he gets trapped in the brambles, and he goes blind and then wanders throughout the forest, blind, trying to find his way home. And they never tell you this in the fairy tale, but presumably he dies because he can't see anything. So this is the fairy tale, but basically, you could replace Rapunzel with Rubisco. Rubisco in C4 plants is found in these protected cells, the bundle sheath cells. So the tower is the bundle sheath cell. And the brambles are the layers of lignin around the bundle sheath cell that keeps things from getting into the tower, essentially. The knight, ultimately, is carbon dioxide. Carbon dioxide is trying to get to Rubisco because carbon dioxide needs to get to Rubisco so it can ultimately be incorporated into glucose, but it can't get there because this cell is protected. Well, why is it protected? Well, you can also think that maybe there's another knight hanging out somewhere, also trying to get to Rapunzel, and this knight is oxygen. He also wants to get to Rapunzel, wants to get to Rubisco, because Rubisco interacts both with carbon dioxide and oxygen. But for carbon dioxide, Rapunzel has a way of getting past the defenses of the tower. Rapunzel's long hair is PEP carboxylase, which picks up CO2 and carries CO2 to where Rubisco is. And Rapunzel's hair is never offered to oxygen because PEP carboxylase doesn't interact with oxygen. So this knight here is basically left in the cold, unable to get access to Rubisco. I don't care how you learn it. All I care is that you learn it. And if it takes going back to a fairy tale and building an analogy for it, if that works for you, great. If it doesn't work for you, find your own analogy. If analogies don't work for you, find some other way of doing it. Graph it out, draw it out. Any process that you can draw from scratch without looking at a book or your notes or anything else, if you can draw that, you know everything there is that we need to know about photosynthesis, C3 and C4 photosynthesis. Figure out a way of getting this socked in your brain. Lots of different ways of going about doing it. What matters is the end goal, not how you get there. Rapunzel. Like any analogy, it has its flaws, because let's face it, a knight who's named Oxygen could just as easily climb up that strand of hair. But of course, in this case, it can't, because that strand of hair isn't really a strand of hair. It's actually PEP carboxylase. So, why are we going into this level of detail about C3 versus C4 photosynthesis? I gave you the reason the other day why we're doing this. It goes back to stuff in the Omnivore's Dilemma, right? When you read the Omnivore's Dilemma, did you pay attention to the section where they talked about stable isotope analysis? Colin was talking about this guy that he knows who runs stable isotope lab, and he can run tissues through his lab that basically tell you what kinds of isotopes are, for example, in your fingernails. What are the isotopes of carbon again? Carbon has 6 protons, 6 neutrons, and presumably 6 electrons. So normally we have, most carbon out there is what type of isotope?
C6.
Not C6, it's the atomic mass, basically. C12, but we also have C13, and we also have C14. What's the difference between C12, C13, and C14?
Neutron.
the number of neutrons. Each of these has an additional neutron relative to the others. So this is 6 protons, 6 neutrons, 6 protons, 7, 6 protons, 7 neutrons, 6 protons, 8 neutrons. So these are heavier. They weigh more. Well, as it turns out, normally these differences in mass of these atoms don't really matter very much. But at the cellular level, inside of a plant doing photosynthesis, guess what happens in terms of which carbon atoms get picked up first?
The lighter one?
The lighter ones get picked up first. So in a C3 plant that doesn't deplete its carbon dioxide all the way down to 0, the carbon dioxide molecules that it picks up are the ones that are lighter. And so C3 plants tend to be more represented by carbon-12 in their tissues because it picks up the lighter objects first. The lighter carbon first. C4 plants, they deplete their carbon dioxide down to practically nothing. At the beginning of photosynthesis, in this process, when there's a lot of carbon dioxide, C4 plants are also picking up the lighter carbon dioxide molecules first. But then once it's picked up all of those, the only thing left are the heavier isotopes. But it also picks those up because it continues doing photosynthesis until the carbon dioxide is mostly depleted. So C4 plants have more of a representation of carbon-13 and carbon-14. These are the rarer isotopes of carbon, but C4 plants have more of those in their bodies because of the way in which they do photosynthesis. So if you eat a lot of corn or a lot of sugar, both corn and sugarcane are C4 plants, you're going to have more C13 and C14 in your bodies because you're eating the bodies of plants that picked up more of these isotopes when they were doing photosynthesis. So when Paulin says in his book, for whatever reason, and then doesn't provide the reason why you can take your tissues and see how much of your diet is made up of C4 plants rather than C3 plants, it isn't for whatever reason that nobody knows. We know exactly why you might have a different isotopic signature if you have a diet that is rich in C4 plants. This is why. The fact that Paulin doesn't mention it in the book doesn't mean that we don't know what the answer is. He just didn't take the time to inform you of what the answer is. Now, a casual reader would read over that and just gloss over the fact that I have no idea what he means when he's talking about stable isotopes. I have no idea what this guy does in his lab that allows him to detect what stable isotopes are in organisms. And I have no idea anything about photosynthesis that informs me at all about what Paulin has just told me. And this is the unfortunate thing about writing a book for a lay audience, is you can trot these things out, say, for whatever reason we really don't know, is kind of the implication, without ever going into, well, we really do know, but it's more complicated than I want to take these pages to tell you about, because quite frankly, I want you to buy the book and read the book and enjoy the book. And so, but this is the reason for those statements. We are corn walking, so this is in the section of the chapter called corn walking. We are corn walking because if you take your fingernails or your hair, snip it off, send it to a stable isotope lab, they're going to find that you are rich in C4 plant because you consume a lot of corn-based products in your food chain. You eat corn, you drink corn when you drink a soda. You eat corn when you eat a loaf of bread, because if you go to the store and look at the ingredients in bread, you will oftentimes find high fructose corn syrup is one of the ingredients. High fructose corn syrup is this stuff that they started producing back in the 1970s. Because of this abundance of cheap corn, you can break that corn apart and convert it into sugar in the form of high fructose corn syrup. It was first used in soda. This was a big boon to the soda industry in the 1970s. But now you would find it in stuff like bread. Have any of you guys ever baked a loaf of bread? Did you put sugar in it?
I have, it was like in fifth grade, but like, I think so.
Yeah, who else? Somebody over here? Yeah, you've made bread? What do you put in your bread?
Yeah, I think we put like, it was like in baking class in high school, so we put breads, like flour, yeast, salt, that type of stuff.
Yeah, flour, yeast, and water is basically all you really need. You can put other stuff in there, but I like baking. I like fresh bread. I make ciabatta and focaccia. If you've ever had a focaccia pizza, like making some focaccia bread and using that as the base for your pizza dough is the best pizza. So my wife got me a bread baking book. It's called the Bread Maker's Apprentice, I think is what it's called. It's a big thick book. It has all these recipes and it's got all of this advice and wisdom from this guy who's been baking bread for like 40 years. And it's really good. You use his methods and your bread just magically turns out to be better than it was when you were making it before. And a lot of it has to do with the time that you put into it. You have to mix it up on one day and then you let it sit overnight and then you do something to it the next day and then you let it sit overnight and then you do something with it the third day. So if you want bread on Thursday, you better start making that bread on Monday. But it's really good. And you know what hardly any of the recipes call for? Any form of sugar. Yeah, I challenge you, next time you go to the grocery store, pick up 10 loaves of bread, different loaves of bread made by different companies, and in almost all of them you will find high fructose corn syrup as one of the ingredients. Why? If it's not necessary to the bread making process, why is high fructose corn syrup in the bread? Yeah.
It's probably like a cheap way to add flavor without putting in like the extra days, like you said.
So it makes the bread slightly sweeter. What do we know about people and sweets?
Like,
We love them. We love them. So if I put a little bit of high fructose corn syrup on my bread, it gives it a sweeter taste, it will make the consumer want to eat it more. You eat more bread, you go through your bread faster, you have to go back to the store and buy more bread. It's a simple equation. To put a product in something like bread that is actually totally superfluous to the bread making process, there's got to be a reason for it. The reason for it is to get you to buy more bread. So even if you're eating things like bread, you're getting some corn in your bread, even though it's not really called for. You're getting it in the form of high fructose corn syrup. So corn has seeped into our diet in all of these ways, such that if you look at our stable isotope signature, we look like Mayans who eat a lot of corn because they grow a lot of corn. And they eat things like corn tortillas. How many of you like corn tortillas? How many of you prefer corn tortillas to flour tortillas? Okay, a couple of people prefer corn to flour. Most Americans, when they go and get a tortilla, they get a flour tortilla. Smoother texture, probably tastes sweeter because there's probably some high fructose corn syrup in there. But people who eat corn tortillas on a regular basis have a similar stable isotopic signature to a Midwestern American. Because corn has crept so far into our food system, not because we eat a lot of corn, but we eat a lot of products that are derived from corn. So that's what he's talking about when he says, we are corn walking. Because we intake corn in soda, we intake corn in bread, we intake corn in any packaged good, really. But we also get corn from our meat, because if you go to a concentrated animal feeding operation, they're feeding those animals not on pasture, they're feeding them in a feedlot, they're feeding them on grain, and a lot of those grains are corn. So if you look at the stable isotopic signature of a cow or a pig or a chicken that is raised in one of these concentrated animal feeding operations, you will also find that they have a strong isotopic signature that represents a diet full of corn. So then when you eat the chicken or the eggs, or the steak, or the hamburger, or the pork chop, you're getting corn's carbon. It's just been cycled through another animal before it got to you, the animal that is finally eating at the end of the food chain. And so this is why Holland says we are corn walking, and it's because of C4 photosynthesis. A long walk to get you to understand like 3 paragraphs of a book. But if you don't have this knowledge, those 3 paragraphs don't mean hardly anything to you. So over the weekend, go back and read those paragraphs where he talks about stable isotope analysis, because hopefully you will read those pages with those paragraphs with a little more understanding about what exactly he's talking about. Chapter 1 is difficult because every section of chapter 1 has a different form of science in it. If you don't understand the science underlying what he's saying, those things don't really mean as much to you as they would if you did have the science. Questions about the basics of C3, C4 photosynthesis? Think about ways of studying this that gets it socked in your memory. If you're still having trouble with the 3 or 4 ways that I've shown you how to kind of handle this, come by and see me and we'll come up with some other ways in which you can maybe incorporate this into your brains. Now I do need this. Theory. There's up. Okay. This thing had a screen on it earlier, right? Did it? I thought it did when I came in. I haven't really looked at it since then. Oh, it clicked off. So I've given you this graphic to kind of illustrate this. I've given you this animation to illustrate what happens in a C3 plant. Once again, kind of reviews, it goes through what we reviewed on the board earlier. This is a diagram of a C4 plant showing you the mesophyll cells and the bundle sheath cells, showing you malic acid as the transporter. Another way of thinking about this, if you want to think about an analogy, is you could think about the bundle sheath cell and the mesophyll cell as being 2 countries, and there's a border checkpoint at the boundary between those two countries. And in this case, malic acid is the semi-truck that carries carbon dioxide from one country into another country. There's a bunch of ways that you can think about it. Once again, pick one that works for you and run with it. There are a couple more images in here that kind of break it down for you. C3 plants versus C4 plants. This is the reason why plants don't want to come into contact with oxygen. And it's because in regular carbon fixation, Rubisco interacts with carbon dioxide, adds it to RUBP, you get these 2, 3 carbon molecules, and eventually those get stripped off and form glucose. So it has to make 6 turns of this cycle in order to make 1 molecule of glucose. In this case, you're going through this process, but you pick up oxygen that gets added to RUBP, and it only makes 1 of those molecules. And as a result of that, you get half of the amount of carbon dioxide being fixed, because it still produces glucose, it still produces PGA that goes into making glucose, but you're getting less of it each time. So you still make glucose, but it's like way less efficient than if you're just fixing carbon dioxide. And so photorespiration uses more energy and also produces less glucose per unit of energy that is being used. And so it's what we call photorespiration because they end up using a lot of energy to make glucose, whereas the regular carbon fixation way uses considerably less energy. So plants are trying to avoid photorespiration. C3 plants avoid photorespiration by just opening their stomata a lot so that they get a lot of CO2. C4 plants avoid photorespiration by separating the making of glucose from the capturing of carbon. Making of glucose happens in a low oxygen environment. Making of carbon, or capturing of carbon, happens in a high oxygen environment, but with an enzyme that doesn't interact with oxygen. So, I've already told you this, C3 plants are favored in cooler climates, C4 plants are favored in warmer climates. So if you look at the temperature and you look at CO2 concentrations, C4 grasses are favored in places where it's hot, C3 grasses are favored in places where it's cold. Just some real world data. If you go up mountains, from the base of a mountain to the top of a mountain, you'll notice that the climate gets cooler and moister as you go up. So if you go out to Colorado and you're down in the foothills of the Rockies, it's hot and dry. As you go up the mountains, it gets cooler and moister. You go from kind of scrub desert into fir forests at the higher elevations. If you are in Argentina and you're going up into the Andes, and you look at what proportion of the plant community is made up of C3 and C4 plants, you find that at low elevations where it's hot and dry, C4 grasses dominate, C3 grasses are relatively rare. As you go up in elevation, they become more evenly distributed, and as you get higher in elevation, it's dominated by C3 grasses rather than C4 grasses, once again because of this climatic change as you go up mountains. Just so you know that this is not a one-off thing, it's a general feature. If you go to Hawaii and go up, I forget if this is Mauna Loa or Mauna Kea, I think it's Mauna Loa. At the bottom of the mountain, you see that C4 grasses dominate and C3 grasses are relatively uncommon. You get up to mid-elevations and there's flip occurs, and you get to higher elevations and it's dominated by C3 grasses, and C4 grasses are relatively uncommon. And it's because at low temperatures, C3 grasses are actually more efficient than C4 grasses because they can just open up their stomata whenever they want, and they don't take the energetic hit that these extra steps in C4 photosynthesis cost the plant. So remember that C4 plants have to spend more energy because they have to basically pick up carbon dioxide twice. One by carboxylase and once by Rubisco. Also, they have to spend energy to transport malic acid out of the mesophyll cells and into the bundle sheath cells. So there's more energy involved in C4 photosynthesis. So if you can avoid C4 photosynthesis, it's a good thing to do. And so C3 plants at cool temperatures actually do more carbon uptake than C4 plants do at those temperatures because they're spending less energy. But as soon as the temperatures start to get hot, C4 plants end up being much more efficient than C3 plants because even though it costs them a little more energy, they're not losing all of this water and wilting and dying in the process. And so temperature really does matter where this is concerned. If you look at in North America and you look at the proportion of the plant community and natural communities that is made up of C3 and C4 plants, 80% of the plants down at the Texas-Mexico border are C4 plants. If you move north, 60% C4 plants, 40% C4 plants, 20%, until you get far enough north, there are basically no C4 plants to be found at all. This is reflected at a continent-wide scale. You see C3 grasses in the montane west, and in the northern part, you see C4 grasses in this hot part in the center of the country. And the whole point, the other point, in addition to understanding what Paulin is talking about when he talks about stable isotopes, is it helps us understand crop distribution. So this is the distribution of wheat in the United States in 2008. And you see that there's a bunch of wheat being grown up in Montana. The first day of class, I showed you pictures from Montana, all these wheat fields that I was bicycling through. Those were in Montana. You also see wheat concentrations down here in western Kansas, western Oklahoma, and the Panhandle region of Texas, which is weird because I think of those places as hot, dry places, not suitable for growing C3 plants. On the other hand, when you look at corn, corn is further south and over a little bit into areas that are hotter and drier. So my question for you, this is a C4 plant in hot, dry areas. This is a C3 plant that is in cool, moist areas, but also in hot, dry areas. How do they manage to grow wheat in western Kansas so easily, given the climate is not suitable to C3 plants? What would you have to do? What would you have to have access to in order to grow wheat in a hot, dry area like western Kansas? How many of you have been out to western Kansas? It's hot, it's dry.
Lots of water.
You need lots of water. Where are you gonna get lots of water?
Either rain or underground.
It doesn't rain there, it's underground.
Yeah.
So this is the distribution of wheat. This is the Ogallala Aquifer. And you see right here in Kansas, Oklahoma, the Panhandle of Texas, underground there's this huge aquifer, and I think it's the largest aquifer in North America, and that aquifer is basically being drawn on to basically allow farmers to plant wheat